All right, I've somehow lost the screen share. Is it still showing for everyone else?
I can see Nathalie's slide, yes.
Okay, cool. All right, I'll just go ahead and introduce her
Okay.
and then yeah. But welcome everyone. So this is the latest installment of the SSC initiative by SSC GSAC to highlight the recipients of the Lewontin Early Award. And it's my pleasure to introduce Nathalie Alomar, who is a third year PhD candidate in Dr. Martha Munoz's lab in the Department of EEB at Yale. She did her undergraduate degree at the University of Florida studying salamander skin bacteria, and she's currently interested in ecophysiological evolution, adaptive radiation, and herpetology. And with that, I will let Nathalie begin her talk.
Thank you, Kathy, and thank you everybody for coming to this talk today. Big thank you to SSC and the SSC Graduate Student Advisory Committee for putting this together. It has been great getting to hear about everybody's work in the series, and today I'm excited to talk to you about my ongoing dissertation work on woodland salamanders. So as a roadmap, I'm going to start by talking about the background and some of the conceptual framework behind my work. Then this will lead me into my guiding questions, which my study system of salamanders will help me answer. And I have some initial results from this project that I would like to show with you all today. And then I'm going to end with the proposed future work, which a lot of this will be able to be accomplished through the funds from this award. So I'll start very briefly on kind of how I got into this research. And a lot of this started in my undergraduate degree at the University of Florida. So while I was there, I worked under a wonderful advisor, Dr. Ana Longo, and her lab primarily researches amphibian disease ecology. So I focus on chytridiomycosis, or chytrid, and this is a severe skin disease in amphibians. It being a skin disease, my work aimed at understanding how the number and abundance of skin bacteria in salamanders reflected their microhabitat usage, and how those skin bacteria were then able to inhibit the chytrid fungal pathogen. And what I found were that species from a relatively warmer and wetter microhabitat harbored this lower diversity of bacteria, yet had the most inhibitory activity compared to the other two species that I was looking at. Now, my time in the Longo Lab was really just a great introduction to research, to academia, and fieldwork, and it gave me a really important first view in how an organism and its pathogen interact with their environment, and learning about how minor environmental changes seem to influence the salamander or any organism, even on a microorganism level, made me think deeper in how an organism can be influenced by its environment. And importantly, it also formally introduced me to salamanders as just an animal, like this redback salamander that you see on the screen. And that really piqued my interest for a lot of other salamander work. As I moved on to graduate school, I wanted to combine this idea of how an organism interacts with their environment through physiology, but importantly, with this unifying evolutionary standpoint. And again, I became very intrigued by the salamander system itself. So over the last 2 years, I've worked in Dr. Martha Munoz's lab, whose lab works on discovering just processes that shape the diversification of reptiles and amphibians. And I've been able to go out into the field and see these salamanders in their natural habitat, which has been amazing, as well as conduct some experiments back in the lab. And today I hope to convey some of what I've learned and done over the past couple of years. So I want to start this talk by thinking of factors that influence the diversity that we see in species. And we can think of 3 key dimensions of diversity: species richness, ecological diversity, and phenotypic diversity as well. And each of these features can be increased or decreased across the lineage history, kind of like changing a dial. So on one end, we can think of this extreme example where we have high species richness, high ecological diversity, and high phenotypic diversity on all 3 of these dimensions. And this is what we see observed in adaptive radiations. An example of this is the anole radiations. And much of the diversity of the anoles arise from the development of ecomorphs. So in the anole radiation, body size and relative limb length of anoles are strongly correlated with structural microhabitat use. So for example, as you can see on the screen, the crown giant has this relatively larger body size, longer limb length that allows it to occupy higher up in the canopy compared to the twig anole that has the smaller body size, shorter limb length that allows it to navigate these distal end of branches. Another example of adaptive radiation are going to be cichlid fishes, and they diversify across many different phenotypic axes, like differences in coloration, body size, pharyngeal jaws, and a number of other features, mostly related to their microhabitats that the species occupy. We can just see a really nice example of all of this diversification right here. Now, another type of way that we can carve up diversification is through non-adaptive radiations. So non-adaptive radiations are these curious counter cases of adaptive radiations, which again have the same high species richness, but conspicuously limited ecological and phenotypic diversification. An example of this is the Batrachoceps salamanders in California, where we see this phylogenetic diversification with relatively limited phenotypic and ecological diversification. And another example of these non-adaptive radiations are these Achentinella snails, in Hawaii. And again, they have these high phylogenetic diversification with relatively limited phenotypic and ecological diversification. A lot of this is a allopatric speciation in these snails specifically. Now, there are many ways that we can carve up these three axes of diversity, like repeated parallel speciation, sexual radiations, and all of this can truthfully be a talk in and of itself. But for my purposes, I'm going to be focusing on non-adaptive and adaptive radiations, and specifically how we infer each of these different types of radiations. Now, when we infer these radiations, a lot of emphasis has been focused on morphological differentiation, whether that be the great morphological differentiation that we see in the anole lizards or the lack thereof that we see in the Batrachoceps salamanders. And this emphasis on morphological features can sometimes neglect the fact that species radiate along multiple phenotypic axes. And these patterns and rates of evolution are often going to vary among trait types. So essentially, where divergence in one phenotypic feature may be inert or slowed among a species, other phenotypic features could be more liable and diversify rapidly. And we have seen this happen before, even in this classic case of adaptive radiations, the anole lizards. So in these lizards, they're ectotherms, right? So thermoregulatory behavior is going to be an essential part of maintaining their preferred temperature. And the anole's ancestral trait is arboreal vegetation. So they're going to thermoregulate through just basking on a tree. And this works perfectly well at low elevations. They're happy, everything's good. However, at higher elevations, thermoregulating on a tree is just not sufficient enough to maintain their preferred temperature. What we see them do instead is thermoregulate on boulders at high elevations, and this allows them to again maintain that same preferred temperature. And this switch from thermoregulating on a tree to thermoregulating on a boulder has been shown to slow the rate of physiological evolution. So as you can see here, we have temperature on the x-axis, performance on the y-axis, and the lighter blue is going to be the high elevation species, and the darker blue is the low elevation species. And we see that the thermal preference is essentially the same in both of these species, again emphasizing this slower rate of physiological evolution. Now, at the same time that the switch onto a boulder slows their physiological evolution, we still see a faster rate of evolution in their morphology. So here we can see how the high elevation species is having this flatter skull and shorter hind limbs that allows it to occupy these boulders and get to their preferred temperature, compared to the low elevation species that need a different type of morphology to bask on the trees. So just in this one example, we can see how depending on which phenotypic trait you consider, the rate of evolution may be relatively slow or relatively fast. So to briefly summarize, we see that morphological traits are the primary trait of choice when considering radiations of any kind, but we know that even in these species-rich adaptive radiations, there can be both stasis and divergence in different trait dimensions. And this has led to my primary guiding questions for my work of how biased are inferences about diversification based on the traits that we examine. And if we're looking at certain traits, does that mean they're independent or dependent of another trait? And to get at these questions, I'm using a very well-suited system, Plethodon salamanders. So as an introduction to our system, salamanders are a part of, our sister to anurans are frogs, and they're part of class Amphibia, which also includes the lesser studied caecilians. And if you were to look at the species richness of salamanders across the world, you'll see that we have this high species richness right over here around the Appalachian Mountains, more so than anywhere else. And this is where a large chunk of my fieldwork actually takes place. Now, the family that I work in is Plethodontidae, or lungless salamanders. And this lungless feature is going to be really important, and I'll talk about this in just a little bit. But this family accounts for 2/3 of salamander diversity. Now, they're primarily found in the Americas, but we do have a few lineages in Europe and even a species in Asia, which is super cool. And these are a few examples of just the beautiful diversity that we see across the family tree. There's differences in morphology and the developmental biology, the environments that they inhabit, and so much more. And even we can see this some species from the Blitogosa genus who use the prehensile tail to hang from trees, which is truly amazing. But my work is going to be focused on the Plethodon genus that you see highlighted here. And the Plethodon genus are woodland salamanders, and this is specifically going to be from the eastern United States, which is where the majority of the species are. There are a few in the western United States, but all of my work be focused on the eastern Plethodon. So here's a few examples of the species that I work with, Plethodon salamanders. So these are terrestrial woodland amphibians. Depending on how the day looks, you might find them underneath like a large rock. You can find them under a large log, or if it's really good conditions, which would be just like a very rainy night, you might see them kind of walking across the forest floor that you see here. And you can see an example of kind of what these environments look like in this lower left-hand corner. So we're looking at the woodlands in the Appalachian Mountains. Now, the Plethodon salamanders can be split into 3 different sublineages. We can start first with the Plethodon cinereus complex that you see here in the red node, and this includes the redback salamander that I mentioned in the beginning. The blue node has the Plethodon welleri complex, and the green node with the most amount of species is the Plethodon glutinosus complex, commonly known as the slimy salamander. And this tree is going to be the tree that I'll be using when I showcase my preliminary results, and this tree does a really good job at dividing between the main sublineages, but there are still some species delimitation issues with closely related species that I'm hoping to tackle with my work as well. So these salamanders are also a classic case of highly cryptic species. And this has caused some initial problems of identification, like, decades ago. And several of these species were first identified through genetic analysis alone. So for example, I have these 6 species kind of up on the board, and you might assume that these are, just by looking at how they look like, that these are 2 species, right? So we have like the top group that looks pretty similar and the bottom group that also looks pretty similar. But what we now know through molecular analysis is that these are actually 6 distinct species. They just have very minimal morphological variation. Now, despite having this minimal morphological variation, this clade has a paradoxical high mix of species richness, especially for members in the glutinosus clade, and they have this really rapid species diversification compared to the other 2 sublineages. And this puts itself as just a textbook definition of a non-adaptive radiation. And so this is really perfect for me because there are several features about this clade that encourages a deeper look at its radiation and to more rigorously investigate the idea that this is a non-adaptive radiation. And I specifically want to focus on their microhabitat preferences and the physiology of these salamanders. So we'll start with the microclimate preferences. On the right, I have a tree and it's showing their preferred temperature across the sublineages. So a color of red is going to be a hotter or warmer temperature, and blue is going to be a cooler temperature. And as you can see, there are different temperature preferences within the glutinosus sublineage that's preferring these warmer temperatures compared to welleri and the cinereus sublineages that are kind of preferring these temperate and cooler temperatures. And what's another important thing to note is that they're not all preferring the same temperature within their sublineages. So for example, for glutinosus, we're seeing a lot of variation in the temperatures that they're preferring, and we're seeing a lot of variation between and within sublineages. And we see the same thing happen when you look at their preferred humidity. So again, we're seeing differences between each of the sublineages. So glutinosus is preferring kind of this wetter humidity, So values of blue are basically showing a more wet environment or a cooler, with a cooler temperature or cooler humidity, excuse me, and red's going to be a more drier condition. So again, we're seeing variations within and between sublineages. And a natural next question is, are these species in any way adapted to these microclimate preferences? And how might that be reflected in their physiology? So are the ones that are in this warmer and wetter environment that they prefer differ from those in these cooler and moist and cooler and drier ones? So let's first explore what that would mean for their hydric physiology. So to start, amphibians' water loss rates has been a pretty active area of research for a while, and we can think of two non-mutually exclusive ways of how these water loss rates may vary. And understanding this variation across the whole plethodon clade is going to be important because plethodon salamanders are completely terrestrial. So they completely live their whole life on land. They have no aquatic developmental stage, which is different from other members in their family. So their hydric physiology is going to play an important role in ensuring that they don't desiccate, that they don't dry out. And the first factor we can think of that's going to play a role in water loss rates is body size. Because again, despite this low morphological differentiation, we do see that body size varies between some species of plethodon. So having a smaller body size is going to result in a higher surface area to volume ratio, which can lead to higher water loss rates. Compare that to a salamander that might have a relatively larger body size. It's going to have a higher surface area to volume ratio or a lower surface area to volume ratio and lower water loss rates. Now, it's important to note that morphology is not the sole factor influencing water loss rates, and that the salamander's environment can also have an impact. So for example, we can think of salamanders at lower elevations, and this is also an important point to consider, considering that these are mountain salamanders, right? They're found across the Appalachian Mountains at varying elevational ranges. So understanding how they might adapt to different elevations is going to be important. So again, salamanders at lower elevations, it's going to be warmer there, it's going to be drier there, so they might be more desiccation tolerant. Compare that to salamanders that are in higher elevations, which is going to be colder, it's going to be moister, so they might be less desiccation tolerant as they are not adapted to these high desiccation conditions. And the question might then arise if there's a potential concern for desiccation or drying out, because again, fully terrestrial salamanders, why aren't all of these salamanders desiccation tolerant all the time? And this leads me to my next physiological trait of interest, which is going to be metabolism and respiration. And what we see is that there's this trade-off, this delicate balance between respiration and desiccation tolerance. Again, these salamanders are completely lungless, so they rely on skin respiration. You can kind of think of it as them wearing lungs on the outside. So as a result, they require a moist surface for oxygen to absorb, just like we do in our lungs. So consequently, having these higher water loss rates, which again could be potentially detrimental if they're in a very environmentally challenging environment, but it can also lead to increased respiration. On the other side, if they have these lower water loss rates, which again can be really beneficial as they don't have to worry about drying out as much, but it could also decrease respiration. And this trade-off has been observed in several species within this clade, particularly in more challenging environmental conditions. However, a comprehensive look at water loss rates and metabolic rates has yet to be done across this whole clade. Now, outside of moisture, temperature is going to play a big role to these ectothermic animals, especially these critical thermal limits. So we can understand critical thermal limits by observing this thermal performance curve. So here we have two axes. In the x-axis, we have body temperature. In the y-axis, we have relative performance. And what we see is that there's this almost normal distribution relationship that many ectothermic animals have with temperature. So at some ideal temperature, they have their highest performance or the optimal temperature. But this whole graph is bound by these upper and lower critical temperatures, so CT min and CT max. And those values represent the temperature where an individual loses coordinated muscle function and is no longer able to right itself if you were to place it on its back. And it could be likely close to its lethal limits. So this is going to be a crucial feature to consider and measure across this whole clade. So in summary, this group is a key system to consider this multidimensional perspective of phenotypic diversity. due to the fact that first, they have these microclimate preferences that has been shown to vary across this lineage in many different ways, and that their physiology hints at different areas of potential diversification from research that has been done in a couple species. But in order to really explore this question, I need to collect a good amount of data that I have been collecting and I'm still continuing to collect. So I'll go through some of the data that I'm collecting now. And I'll start with the environmental data, because in order to contextualize any physiological patterns or morphological patterns that I may observe, I need to understand the environment that they're in, including factors that may influence temperature and moisture. So when I go out there, I'm collecting relative humidity, dew point, the wind speed, elevation, of course, soil temperature, air temperature, and the substrate that the salamander is in. So it's underneath the log, it's underneath the rocks, it's just in the leaf litter. And this allows me to really identify the microclimate that the salamander is in. And this will look something like this. So here's a few photos kind of showing what the fieldwork looks like. We're normally going on trails in the mountains and looking at salamander or looking under rocks in areas that we're gonna find salamanders. And normally it looks a lot darker than this because we're doing nighttime herping or nighttime collecting, since these salamanders are completely nocturnal. So it's a completely different activity levels that you see if you go in the daytime versus the nighttime. So it's really just us salamanders and headlamps, which is always nice. So once we have these salamanders, I bring them back into the lab and they go through just a nice little trial of physiological experiments. So we have water loss rates, metabolic rates, and 3 thermal physiological traits: CT-min, CT-max, and T-pref. Now for CT-max, how we do these experiments, again, remember that these critical thermal temperatures or an animal loses its locomotion function to right itself. So what this looks like for the CT-max experiment is putting the salamander in this little water bath, and we have a heat lamp on top of it, and we're very slowly going to heat up the water. Then we start flipping the salamander onto its back until it can no longer flip itself over. Now for CT-min, this is something that's very similar, except we put it in a falcon tube because we're going to add ice into the water bath. And again, we're just going to slowly decrease the temperature. And so we're going to flip the salamander in the falcon tube until it can no longer right itself. Now, for the thermal preference experiment, it's a little bit easier on the salamander. We essentially have this gradient of temperatures, and we have a completely moist environment where they can walk and decide what temperature that they want to be in. And then over the course of about 2.5 hours, we're measuring the temperature of the salamander in this time. Now, the thermal physiological experiments are a little bit more straightforward, but I do want to take just a quick second to walk through how we collect our water loss rates and our metabolism or gas exchange rates. And to do this, we're going to use a respirometry machine. And we start with controlled airflow going through the machine that is then saturated with water vapor pressure, as you can see here. And then that water vapor pressure is regulated by a specific dew point. And this essentially allows us to control how hot, how wet or dry the air is. After that, it's going to be broken into 8 different tubes. So 7 of these are going to have salamanders, and 1 of them is just going to be an empty tube for baseline. And all of these are going to be in a temperature controlled chamber. After that, each of these chambers are read 1 at a time over the course of 3.5 hours, including our baseline. And from there, we're able to collect our water vapor pressure and our percent CO2. That is then we can extract using a software called Expedata and convert into water loss rates and metabolic rates. And what this essentially just allows us to do is get really precise measurements of water vapor and percent CO2. But it also allows us to control for the temperature and vapor pressure deficit, which is just how dry the air is. So when we're doing our water loss rates experiments, we run them at 3 different temperatures and 2 different vapor pressure deficits. So we have from 13, 17, and 23 degrees Celsius. And then the 2 different vapor pressure deficits is essentially a drier condition and a wetter condition. And this allows us to get a broad range of environmental conditions and to understand how they're losing water and if it changes in these different environmental conditions. And then for the metabolic rate experiments, we're doing them just at 1 temperature because we just want resting metabolic rate. We don't want them to be dried out or anything like that. We just want to know what their resting metabolic rate is. Now, we're also, of course, going to collect morphological data, since these can also be tightly correlated to physiology. So I'm going to be taking all of these linear measurements that you see here to describe body size and also body shape. and this will be a relatively more straightforward process than the physiological data. Lastly, I'm also collecting tissue samples from all these salamanders, and we'll use phylogenetic analyses to derive a more robust modern perspective of speciation in this radiation through ultra-conserved elements, or a UCE phylogeny. And what all this allows me to do, to kind of summarize all of this work, is build this large data set that includes environmental data, physiological data, genomic data, and morphological data to really explore these woodland salamander radiation from a multidimensional perspective. And this will allow me to answer this broader question of, is ecophysiology a quote-unquote hidden dimension of phenotypic diversity in woodland salamanders? So now I'm going to go through some of my preliminary results that I have. I'm going to start with our thermal physiology, in particular CT max. So as a reminder, CT max is that upper thermal limit for locomotion, or you can just think of it as heat tolerance. So on this trait map, warmer, redder colors indicate a higher heat tolerance, and cooler, bluer colors are going to indicate a lower heat tolerance. And I'll add that for both heat and cold tolerance, which I'm going to show next, we currently have a limited amount of data, and our statistical confidence is pretty low at this stage. But with that being said, phylogenetic signal in heat tolerance is pretty low, and we're starting to observe some variations within the glutinosis clade, which is really the only clade we can make inferences is, as we only have one other species from there. But like we see some differences in the sister species, and it'll be interesting to see how this continues to shape up, if we're seeing any differences continue to see differences with the sister species as we add more data to this. And we see something similar going on when we look at their cold tolerance. So again, for here, a value of red is going to be a warmer temperature and blue is going to be a colder temperature. And again, we have this low phylogenetic signal across the clade. We're seeing some variation within the glutinosis clade as well, but again, pretty low statistical confidence to see if this is going to maintain or not maintain. Now, on the contrary, for our TPREF values, we actually have a lot more individuals and a lot more species here, as most of the salamanders that we have collected have gone through the TPREF experiments. And the majority of the TPREF values, the thermal preference values, tend to cluster around 17 degrees Celsius, and there are a few exceptions of species preferring slightly higher or slightly lower preferred temperatures. And when you look at the raw data, really, a good amount of variation is actually a lot of variation with individuals of the same species. And sometimes in some of these species, they have a very wide variety of temperatures. But still, we're seeing this low phylogenetic signal. It is noteworthy, though, that most species favoring these slightly warmer temperatures are in the glutinosis sublineages. And these are mostly salamanders from the southern Appalachian Mountains, and these are also the salamanders that have been found to kind of prefer these warmer temperatures. So it is an interesting point to see, and again, once we add more data, we can see if these patterns hold. Okay, so now we'll move on to our metabolic rates. So this is the residuals between metabolic rate and body size. So this has been corrected for body size as well. And the warmer, redder color is going to indicate the higher metabolic rate, and the cooler, bluer color will indicate a lower metabolic rate. And we see that the phylogenetic signal is a bit higher for metabolic rate, with species you can see in the cinereous clade exhibiting this lower mass-specific metabolic rate compared to species in the glutinosis clade. And we see that the glutinosis clade is going to have a bit more variation, especially between closely related relatives. So you can see kind of these 6 or so species over there, they have a lot more variation in this mass-corrected metabolic rate. But overall, we are seeing some differences between the glutinosis clade and the cinereous clade. And this is also echoed when we look at the resistance to water loss rates. So that is what you can see here. So here, again, warmer colors are going to indicate species having higher skin resistance. And this means that they're more desiccation tolerant, so they're losing less water. And cooler colors are going to have species having lower skin resistance, meaning they're less desiccation tolerant, so they're losing more water. And skin resistance really accounts for a lot of biophysical differences that might occur between the salamanders. So it's controlled for body mass and a lot of different factors. But we can think of it as having higher skin resistance, just more desiccation tolerant, lower skin resistance, less desiccation tolerant. And once again, the glutinosis clade is exhibiting the most variation. So for example, we can see here in Plethodon catawensis and Ouachita, these are actually the only two species that I have collected so far that are in the Ozark Mountains in Arkansas. The rest of the species in the gluinosis clade are really found in the Appalachian Mountains. So we're seeing that they have like a lower skin resistance compared to the other gluinosis individuals. And again, if we're looking compared to the cinereous clade, which tends to be a little bit lower than what we're seeing in the gluinosis clade. So in terms of the physiological data, we're starting to see a few patterns show in their thermal physiology, but generally not as much variation in the thermal phys. While in the skin resistance to water loss and metabolic rates, we're seeing much more variation among sublineages. And this could be due to potentially the trade-off between skin resistance and respiration that I mentioned earlier today being an important driving factor. However, there are still other avenues that we want to explore, like looking into elevation and air temperature and other different environmental factors that could influence this as well. Now, the last result that I want to show you today is the rate of evolution for the resistance to water loss rate values and a couple morphological features. So here we have this skin resistance to water loss, hind limb length, head length, and forelimb length for a few species that we have. And as you can see, we have found the physiological traits to be evolving at a much faster pace than the morphological traits. Now, of course, there's a lot more to explore here, as I'll describe shortly, and I'm planning to use funds from SSC to complete a lot of this species sampling. And once I have those data, I hope to explore patterns of physiological evolution in greater detail and test for canonical signs of adaptive radiation, looking at diversification through time, rates through time, and other approaches. For example, I plan to use the node height test as one of these, which essentially tests for accelerations or deaccelerations in trait evolution by comparing the independent contrast for a trait with respect to the node height. So a significant relationship can imply that the rate of trait evolution is shifting through the tree compared to more than what we might expect with Brownian motion. And a late or an early burst of evolution are indicated by the sign of the slope of the line that we see here. So a negative slope could be an early burst of evolution. This can imply that a trait evolved rapidly during the early stages of a clade's history, and it starts decreasing as niches become filled. This is one sign that has been used to show that it could correlate with adaptive radiation, and a positive slope could suggest that a particular trait has evolved or increased over time later on in a clade's history. Now, the rate of evolution in looking at the trait evolution is still very preliminary. What I do want to point out is that there seems to be something interesting going on with ecophysiology in the woodland salamander. And I think even with what we're able to observe now highlights that just because one aspect of the phenotype is not evolving very fast, doesn't mean that all of the other traits are inert. So in summary, my current data is pointing to more variation in this hydric and metabolic physiology, especially when we're comparing it to the morphological data. So I'm pretty close, but not yet done with kind of collecting data for this whole data set to be analyzed. And in all of these kind of sections, I still have a little bit more data to collect. And this will help me be able to put to test this idea of Plethodon salamanders being a non-adaptive radiation. And regardless, this is going to contribute to our understanding of ecophysiological evolution in salamanders and mechanisms underlying their very high diversity that we see here in North America. Okay, so for the last part of this talk, I'm just going to kind of talk about what I plan to do with the funds that SSC's got me and update on a little bit of what I have done. So up to this point, I've collected 265 individuals from 25 species, which has been such a roller coaster, but really fun. And as you can see from like all of the data that I've shown, there's still a number of missing species that I intend to incorporate. But because of SSC and the funding from this award, it has helped me and will continue to help me collect more of these species. So right now, or at least at the time of my award, my goal was to collect 14 more species. I got this award in May. And prior to that point, a lot of my collection has been kind of like really strongly in like the southeastern United States, where we have South Carolina, North Carolina, Virginia, West Virginia, and Tennessee. And this is really where we see a lot of diversity of salamanders. And right now I'm trying to collect basically a lot of stragglers. So we have Ohio, Arkansas, Alabama, and Georgia. I've been to some of these states already, but I still need to collect a few species from there. And that's kind of been the main goal. But Virginia and West Virginia, I was actually able to complete just a couple of weeks/months ago. So these are a few of the salamanders that we were able to find. So I've collected so far 7 species from the 14 that I had aimed to collect. And I just have around 7 more, and this is maybe 8 to 10 more days of field work. I'm really hoping to complete this within the next couple of weeks. But a lot of these sites that I have collected from have been from Virginia or West Virginia, and these are very high elevation species. So as you can see here, we're kind of at like the top of the mountains, and a lot of the points that we've collected at have been like the highest elevation in Virginia or West Virginia. So it's been a lot of fun seeing a lot of different species of salamanders and bringing them back to the lab. And now a lot of them are acclimating, getting ready to start kind of like the throw of physiological experiments that they have to go through. And then one thing I do want to mention about at least one of these trips is that I've been able to go with some undergrads as well, which has been really fun. So we take them out to Virginia and let them literally get their hands dirty, flipping logs and rocks for salamanders, which has been really great to have them in the field, kind of like seeing the animal in their natural habitat and really getting them to think of questions related to, evolutionary biology, ecology, and just research in general. That's been just like a really fun time of these last few trips. And I also want to mention that the salamanders that I collect in the field are also going to be instrumental in projects beyond just this one. So like I mentioned, I really want to create this UCE phylogeny. This will be really important for a lot of the comparative methods that I want to do with this work. But on top of just getting this more robust family tree, I also am going to be doing some work looking at hybrids. So we have a few hybrid populations that I've collected over the last kind of year or two. So for example, we have Plethron sherbani, and this is a high elevation species, and Plethron teahali is this low elevation species. And at this mid elevational range, we kind of see this hybrid population that literally looks like a mix of these two, kind of just phenotypically speaking. And because of the salamander's really, really large genome, which I won't get into right now, but they have a very, very large genome, which made it kind of difficult for a lot of these advanced genomic methods. But recent years, it's been a lot of progress in there. So I really want to investigate their genetic background and their ancestry of these hybrid populations. Also, the other fun thing about this work is that we're collaborating with the Riddell Lab that's at UNC Chapel Hill. So research in Eric's lab has shown that without incorporating skin and boundary layer resistant values, estimates of potential activity time and energetics can be greatly underestimated. So you can see these are kind of these estimates without incorporating skin and boundary layer resistance, which look pretty dire. But when you do include skin and boundary layer resistance, you see that we have been underestimating. And this work has been done so far with one species of salamanders, Plethodon metcalfi. But the goal with this collaboration is to include all of our physiological data into these biophysical models, providing a more comprehensive spatial and phylogenetic perspective of the vulnerability to climate warming in this imperiled lineage. And this is going to be really important. These salamanders are an indicator species. They play a very important role in the Appalachian Mountains or in any of the mountains that they're found. So being able to help understand how they're going to be impacted by climate change is going to be super important. And then finally, I just want to end with what I hope my work can do. And I hope like this work on these adorable little woodland salamanders can emphasize that evolution is a multidimensional phenomenon and that our biological inferences about diversification should better embrace and anticipate like these complexities and nuances among these different trait dimensions. And to be very, very clear, my intention is not to say that non-adaptive radiations are adaptive or vice versa or anything like that. But really what I want to emphasize is that our understanding of a system is going to be dictated by the perceptual lens that we apply. And that sometimes, and perhaps often, the inferences that we make could dramatically differ depending on the traits that we examine. And finally, of course, I want to have a bunch of gratitude to all of my funding sources and to the Munoz Lab, past and present, to the Riddell Lab, and really a major, major shout out to a lot of summer undergrad interns that I've had over the last 2 years that have really helped collect a lot of this data and make a big push for it. So big shout out to my undergrads as well. Then I will take any questions.
That's awesome. Thanks so much, Nathalie, for your talk. Yeah, feel free to unmute if you are interested in asking a question, or type it in the chat and we can read it out.
Yeah, I just had some questions. So like I had a methods question about the salamander flipping.
Mhm.
Like are there other things besides the temperature that could influence its ability to flip? Like they use their tails to flip, or do they not? I don't know how they.
No, for the most part, if you were to flip them at just like room temperature, right? Like most animals, they don't like being on their back. You have a lot of vital organs. Like this is just not an ideal position. So they're
Mhm.
just gonna move. And that really has to do a lot of coordination, like with your brain and the muscles and everything like that. And essentially what we see when they're at kind of like their thermal limits, and we do kind of probe them sometime, especially with CT Max, like they might be like this and they won't flip over. You touch their tail, they're like, okay, I'm gonna do it. But at a certain point, they're just like, I'm just too hot or I'm just too cold and I don't care anymore. Like I'm just going to stay on my back. So it really is more like the strongest thing that we see correlated with it is temperature, because they're just not into it.
Gotcha. So as you're approaching those highs and lows, you've already been
Yes.
prodding them.
Yes, for
Gotcha.
like the last couple minutes or whatever. As soon as we kind of know what it will be like, so we just do it, which I'm sure isn't fun to them. But then we know at a certain point they just completely tap out. Not completely.
And I thought
I have
someone
a
had their.
question.
There we go.
Sorry, my lab mates were talking and now I'm in the class. There we go. Sorry, Nathalie. That was an amazing talk as always. I'm always so blown away by all the amazing work you do and how well you communicate it. And several of us noted how you lit up when talking about the salamanders, which is always nice to see. Okay, my question's very broad, so apologies. But I'm so inspired as a microbiologist by how you are trying to look at these salamanders from the organism's point of view to try to find traits that are relevant. So I was just hoping you could just wrap on maybe the importance of like natural history or just like sitting and observing your organism on trying to find important traits to examine for looking for these adaptive radiations.
Yeah, no, that's a great question, Noah, and thank you. That's so kind as always. No, that's a great question. And I think that it's like, right, in the ideal world, you just test everything, right? You just get all the traits possible and then you can really understand it. But that's the ideal world, it's pretty impossible. And I think with a lot of these other kind of classic case of adaptive radiations, morphology is just so obvious, right? Like it's there, like they look different, right? And it's for the physiological stuff, it's easier to actually like measure as well too, if you're thinking about when these adaptive radiations were first discovered. But I think when you think of, even trying to compare like lizards to salamanders, right? There's a lot of differences there. And I think with these salamanders, they kind of provide this good narrative for why that might not always be the case, and kind of just thinking of like, why am I interested in like this trait? Is it because everybody has been interested in this trait? Look to the history of it versus like, how is this actually relevant to the questions that I'm asking, what I really want to understand about the organism in and of itself? And I can definitely say that even with the salamanders, like being out when it's been very, very dry for like the last, I don't know, 2 weeks, it is pretty pathetic to like try to look for them. It's very disheartening versus it rains one time and it's like a completely different atmosphere almost. Like there's salamanders crawling around. Some of them even climb trees if it rains like hard enough, like a little bit, like at least a foot off, which is kind of crazy. So I think it's also being able to understand your organism, really think about why I'm trying to study what I'm studying, and think deeply about the why of your questions. I think it's easy to kind of like just kind of go with the flow. I'm like, oh, I was told to do this, I'm just going to continue doing this as well. But being able to understand like, why am I actually thinking about this? What's the importance? Like, is this really the best way to get at it? I think would be the biggest thing that I've gotten from the work so far.
Thank you so much.
We have a question from Tyler in the chat asking what kind of sample size you're working with for each sublineage.
Oh, yeah. So for each of the sublineages are a little bit different because like I mentioned, glutinosis has the most amount of species. So, and again, this tree isn't the best to really denote the different species. Glutinosis has the most species, and so we're going to see probably more variation just because of the species itself. Cinereus and Wellery tend to have a lower amount of species. But even within, so again, I don't know the exact numbers, but we're dealing with around 10 to 12 of Plethodon cinereus. There will be some species we just can't get because they're endangered, of course. Wellery has a lot less than that. So we're looking at around 8 or so, and glutinosis has, I want to say, around like 16, 18 species. Which is also kind of the interesting point because glutinosis, a lot of these species like diversified very, very rapidly compared to the other two sublineages. So I also wouldn't be too surprised if a lot of the differences that we see or a lot of the driving forces for this diversifications kind of centering around the glutinosis clade. We might not really see a lot in the Cinereus Wellery clade, maybe just because of how many species there are alone or a few other different factors. But so glutinosis does have the most amount of species. Within each of the species, we are getting around 10 to 15 individuals per species as well.
I have a question, especially in species where you're seeing variation among individuals of the same species. Do you have any speculation for why that might be? And also, especially given your prior interest, is there anything known about how the microbiome might contribute to the variation in the physiological traits in these salamanders?
For some of them that have the variation within species, I think a part of it is that some of their range sizes are pretty insane. So like there's this one species, the redback salamander, can be found, like you can find it here in Connecticut, and they can find it like all the way kind of like in this range. And there's some that you can find like in one mountain, right? So some of it could also just be population differences, which I haven't looked at too much, but it is a possibility. In terms of the microbiome, I think that's super cool. I would have loved to, as I've collected this, also swab all of these salamanders and kind of see like what's going on there compared to the physiological stuff. Potentially, I can't speak too much on that. I would love to see how they might differ through it, because I'm sure there could be even more looking into, what bacteria that might be, impacting any water loss rates or anything like that. But no, I haven't looked into that. I would love to do that, but great question. I think about it often.
Cool, thanks.
Am I allowed to ask another question?
Yeah.
This one's more like narrowly focused, but I'm curious, since you mentioned like ranges, like how much of this region are there many salamanders coexisting within a single geographic spot? Are they filling different sort of sympatric niches, if I can say that? And also I'm curious whether they mate in the lab as well.
Some of them do have some sympatrixy that we see in the field. So there's a good chunk of them, especially in the like glutinosis lineage that has a lot of species, where it's like if you're in Alabama, if you go to like the north, it's one species. If you go to the south, it's a different species. If you go to the west, it's a different species, right? So some of them are very clearly kind of broken up by the geography. But there's others, you go to North Carolina, you're going to see not just in like Plethodon genus, but like there's going to be other genuses, Desmox, Eurisia, just this like a beautiful salamander party there. So they do have some symmetry, which is very interesting, and not just within their own genus, but with a bunch of others part of the salamander family. No, they don't mate in the lab. That would be so nice, but they have actually been known to be very difficult to mate and to reproduce in the lab.
Oh, that's a bummer. That would have been really cool, but
Cool. Yeah, I know. Not like bacteria.
I was just wondering, once you have those UCE phylogenies, would you redo your analyses on that one that you built?
Yes, definitely. The tree that I have right now, there's just a couple species that are just not even included into it that we do have in the lab.
Oh, wow.
So yeah, so even just like, I'm adding branches now, but it's just for preliminary stuff, right? So for the UCEs, we really do want to get every single individual and also make sure that we're identifying them correctly as well too, as like a sanity check. So definitely going to be doing everything over with the UCE phylogeny.
All right, well, if that's all the questions we have in the video in the chat, we'll thank Nathalie again. Great talk, very passionate talk, like they were saying about like Noah said, you really did light up with salamanders pictures, especially that tree hanging one. And we will see everybody again on Monday for our next Greg seminar. Thanks so much.
Yes. Thank you, everybody. Have a good day.